Hey there! You've probably heard of stem cells if you are active in the skincare world nowadays. They have been everywhere recently, in more trivial applications such as skin rejuvenation, or larger ones like significant wound healing, for example, burn wounds. So, what exactly are these miracle cells? Stem cells are undifferentiated cells that have abilities to regenerate and help cells around them to do the same, which is why they can do what they are known for. Since they are undifferentiated, they also can evolve to different types of cells. There are different kinds of stem cells based on their source and functionality. Some of these stem cells are superior to the others in terms of how many lineages they can evolve into, known as pluripotency. An example is embryonic stem cells, or ESCs for short, which have great pluripotency. There are also induced pluripotent stem cells, or iPSCs, that have similar pluripotency to ESCs. However, ESCs cannot be used due to ethical concerns about their sourcing from embryos. On the other hand, iPSCs have been shown to have post-usage complications. Now let's look at another different type of stem cells, hMSCs or human mesenchymal stem cells. They were found to have excellent regenerative capabilities and can be sourced from several different parts of the adult human body. Currently, several hMSCs-based products are even available for clinical use already. The findings regarding these stem cells have shown them to be suitable for treatment, but the issue is large-scale production. Furthermore, due to the nature of their usage, they must be processed strictly for the recipient's safety. So, we looked into several methods based on studies published and available online to find the most efficient way to produce hMSCs without compromising their quality. The processing itself can generally be separated into two parts: upstream processing and downstream processing. The upstream processing encompasses everything about the synthesis of the cells, from the ingredients prep up to when the cells have finally taken shape. Meanwhile, the downstream processing talks all about the finishing of the product, separating the cells from other components in the production mixture, making sure that the cells produced are in fact the correct cells and are in their prime, because we do not want any issues with swap cells or anything, do we? You can take multiple approaches for every step of the process, and each might result in different qualities you might target. For our case, we sorted out several settings that you could do to optimize the production of hMSCs. First off, the use of the MEM or alpha MEM and FBS medium provides the best nutrition for cell growth and are most readily available too. And to further create the best growing environment for the cells, the processing should be done in either stirred tank or spinner flask bioreactors with continuous feeding strategy under the following conditions. As for the duration of the process, it depends on the source of the cells we are culturing and the amount of yield targeted. Now onto the other half of the process, which is the downstream processing. It gets tricky as the process changes settings from planar culture into microcarrier-based bioreactor culture in enormous volumes. Every precautions must be taken to ensure that the cells produced are functional and uncontaminated while maximizing product yield. To begin, harvesting can be accomplished through various methods, including chemical, mechanical, and thermally induced detachment. The most effective way appears to be a combination of chemical and mechanical induction, taking about 15 minutes. Washing can be done with a fast PBS rinse, and the techniques do not change substantially. For filtering, normal flow filtering with a bigger nylon mesh membrane filters is a wonderful solution for large-scale production, since it is easy and cost-effective. Afterwards, the ATF technology concentrates them using a hollow filter membrane. They are made for perfusion. Therefore, they will need to be more significant to handle the flow rates required to reduce the volume in less than 8 hours while remaining viable. Now let's jump to analysis. This is the list of tasks that must be performed. It's a lot, but we must ensure the cells are valid and safe for use. Once they are apprehended as suitable, they can be formulated with freezing media containing 7.5% DMSO, 2% bovine serum albumin, and 2.5% polyethylene glycol. Then, the mix can be placed in containers with a vial filling machine. Once filled, the cells are stored depending on their use. If they are going to be used within a few hours, they are treated with human serum and stored at 4 degrees Celsius. If long-term storage is required, DMSO is added as a cryoprotectant and the cells are held at 4 degrees Celsius in the fridge. This combination of processes is theorized to achieve the maximum yield of high-quality hMSCs in a large-scale production. We hope it can facilitate the delivery of safe stem cell therapy to help those in need.